All right. So DNA replication, and we're going to go through the specifics of DNA replication. All right, and how it actually happens. So we know that it's a semi-conservative model that we're looking at, which means the parental strands are going to be used as what? Templates. All right. So the parental strands are going to be used as templates to make newly synthesized DNA. All right. A couple things about DNA replication. It's pretty fast. Okay. Let's consider that a normal human cell has a doubling time, which means it will go from 1 to 2, 2 to 4, 4 to 8 in roughly 24 hours. A normal skin cell will double in about 24 hours. So that means in that time, a cell has to grow to a point where it needs to double, synthesize all the DNA, replicate all the organelles inside the cell, and split. All right, and then be ready for the next split within a day. Okay, so DNA replication, pretty quick. Okay. Also consider that there are roughly 3 million base pairs of DNA in each of your cells. All right, so going through all of that and making sure that that DNA is replicated almost identically to the previous cell. All right, there are some. All right, this is a very concerted process, meaning that there are directions. There are proteins that are involved in this that have very specific roles, and they all work together, okay, to have this function produced. All right, and we'll go through most of those proteins. All right, several of those proteins. There's lots of them. We're going to be concerned with maybe 6 or 7 of the most important. All right. There's always more work that's being done on replication machinery. All right, there's always new proteins being found, new functions for different DNA enzymes, etc. All right, and we will also see some of the differences between prokaryotes and eukaryotes in DNA replication because they are different. Okay, questions up? All right. Where does replication start? All right, so a replication fork. So a replication fork. What is a replication fork?
When the DNA splits.
All right, so splits. Is DNA normally found split?
No.
No. All right, so there has to be a point somewhere along the chromosome, which is double-stranded DNA with proteins in a certain structure. There has to be some place along that double-stranded DNA that is marked as a starting point. Okay? So this is what we call an origin. Okay? This is a starting point for DNA replication. All right? At that point, that double strand, the double-strand structure of DNA becomes unwound. And then that begins what you have as a replication fork. Okay? So we have origins of replication, and they form sort of a bubble so that it looks something like this. Here's one strand of DNA, right? Here's another strand of DNA. And now at this origin right there, okay, they would separate apart. And now you have the beginnings of a replication fork. Okay? So now these two strands are separated apart. And then at other points they come back together. All right? So you have this bubble that's been produced that forms a replication fork. All right? And if you look, there are actually 2 points where there are replication forks, right? So because they're double-stranded and you open up a bubble in the middle, there's actually a replication fork here and here. Okay? And so you can get progression of DNA in both directions. We'll go over that in a second. All right? So, but really we're concerned with this replication fork, all right, where the parental strands become separated apart. What separates? How does a cell separate DNA? How do you separate the double strand? What do you expect will provide that function? A protein, okay? And that's exactly what happens. There is a protein that will bind to DNA and sort of force the two strands apart, okay? But how do you think it knows where to bind DNA? So let's say this. Let's go through what we already went through. So DNA replication starts at an origin of replication, and at that origin, DNA unwinds, okay, and forms a replication fork. And I've now told you that a protein is responsible, an enzyme is responsible for binding to DNA and opening the replication fork. Given those three things, what can you conclude about where that protein binds DNA?
Binds to the origin.
Binds to the origin. Okay? So it has to be able to recognize an origin and open up the DNA at that point. Okay? So sort of a deductive reason. All right? So here's your replication fork. And the enzyme that opens DNA is called a helicase. So DNA is structured in a double helix, right? Enzymes end in ASE. Helicase opens the helix, right? Unwinds DNA and produces your replication fork. Okay? Other important proteins in here. Okay? Now, where that replication fork is, DNA is found in two separate single strands. All right? DNA does not like and is not usually found in that structure. Okay? And in order for replication to occur, DNA has to stay in a single strand formation. All right? So in order to keep it in that single strand formation, you have these things called single-stranded binding proteins. They bind to that single strand area and hold the replication fork open. Because otherwise, if it doesn't hold open, what's going to happen? What's the normal structure of DNA? The most stable structure of DNA? The double helix. So it will flip back together and resume its double helix structure. And that would be counterintuitive or an obstacle for replication to continue. Okay? Questions on helicase, single-stranded binding proteins. We'll get into the other ones in a little bit. Okay, but for now, helicase, single-stranded binding proteins and what they do. You're going to have, you'll see topoisomerase, and right here, there is a nick in the DNA. So think of going back to our rubber band analogy. If I twist the rubber band and I pull it apart, it gets tighter at the end. Now, at one of the ends, if I cut the rubber band, what's going to happen to the stress on that? It's going to be relieved. All right, what it allows is you cut the rubber band, and it's going to rotate around the other band and relieve the stress. Okay? And that's exactly what topoisomerase does. It nicks one strand, and the pressure or the tension is relieved as it rotates around the other strand. Okay?
Does it rebind that strand once it goes down the line?
Does it what's called ligate the strand together? Yes. So this it'll go back together. As helicase moves open, it'll open it back up too. Okay? And there's a couple other functions for topoisomerase, but in terms of replication, this is its main purpose. All right, so we've gone through helicase, single-stranded binding proteins, and topoisomerase. We'll get into what a primase is in a little bit. All right, topoisomerase. So helicase are enzymes that untwist the double helix. All right, single-strand binding proteins, they bind to and stabilize the single-stranded DNA. Topoisomerase is another enzyme that actually acts ahead of the replication fork. All right, so it acts downstream of the helicase. So as helicase is unwinding the DNA, right, it's pulling it open. Ahead of the replication fork, the helix is getting tighter and tighter and tighter. Think about if you had a rubber band and you twisted it over, right? So it's sort of a rope pattern. And then you held it together like that and pulled the two strands apart of the rubber band. What's going to happen to the rubber band at the ends? It's going to get tighter, okay? Because you're pulling that apart. You're causing a lot of stress ahead of the helix. And that's what's happening to DNA. So to relieve that stress, and which will further allow helicase to process down the replication fork, topoisomerase does its function. Okay? So ahead. What is a difference between the genome or the DNA of eukaryotic cells versus prokaryotic cells? How are they structured? What's that? What's found in the nucleus?
DNA is found in the nucleus.
All right.
Eukaryotic.
DNA is found in the nucleus in eukaryotic cells. What is the general structure of DNA in a prokaryotic cell? Are there proteins binding that DNA? No. There's no nucleus, right? There is no structure in terms of chromatin structure, right? Compaction. For the most part, in prokaryotes, their genome is much more simple, all right? It's much smaller and much more simple. In terms of, it's just a double-stranded piece of DNA. Okay? The other thing is that the genome is circular. Okay? So what you have here is origin comparisons in E. coli, which is a circular chromosome, okay, versus what you see in eukaryotic cells, which have larger, much more complex genomes, chromosomes. All right, so if I said the usual eukaryotic, the usual for E. coli, the size of the E. coli genome is about 25,000 base pairs. Okay? Versus what you see in a eukaryotic cell, which is somewhere on the of 3 million base pairs. Which one's going to replicate faster?
Prokaryotic cell.
Due to their simplicity. Okay? So so one origin, it's going to, and I'll say that the enzyme that does this will replicate about 1,000 base pairs per second. All right? So it goes pretty quick. Okay? And what do you know about the growth of E. coli cells?
Goes fast.
All right, it's really fast. So overnight you can have millions of cells, so you have to have some way of replicating DNA rather quickly. All right? Look at the difference between the origin. What do you see between the difference between eukaryotic and prokaryotic cells in terms of the origin of replication between these two? There's more than one. Okay? So the way that eukaryotic cells, one way that eukaryotic cells address their complexity in terms of DNA and their overall size of genome is by having multiple origins of replication along a chromosome. Okay? So in prokaryotic cells, it's okay to start at one point. You're only going so far. It's like, I can start at Hickory Hall and walk to the Union. Okay? All right, fine. We're walking there and back. No big deal. But if I wanted to, well, that was probably a bad analogy. All right, but if I wanted to cover more ground in a same amount of time, I could start at different points. Okay? It's like a relay race, all right, rather than one person running that one distance. Okay? So that's how eukaryotic cells overcome that size problem, all right, in terms of their genome. Questions on that? All right, so multiple replication bubbles form and eventually fuse, speeding up the copying of DNA. All right? So to this point, we have opened up the strand, formed a replication bubble. Single-stranded binding proteins are holding that bubble open. Helicase is responsible for opening, unwinding the DNA. Topoisomerase is ahead of the helicase, relieving any stress that's being formed on the strand. Now we're actually going to begin the process of replication. Okay, that single strand, that replication bubble that's open, that single-stranded DNA is going to provide the template for new base pairs, nucleotides to be brought in. Okay? But the process can't start on its own. The enzyme that's responsible for copying and synthesizing new DNA is an enzyme called DNA polymerase. And there are multiple copies of DNA polymerase that are present, and they do certain types of functions. All right? But it cannot start on its own. Okay? It needs a template, or it needs a starting point. What do you think, and remember back to class on Monday, what do you think is the point that it needs to start at? The 3' OH. Remember that recall that from Monday that I said new nucleotides are added to which end of the strand? That free 3' OH. Okay? If you have a DNA, all right, so here we have one strand of DNA, okay, and the other strand of DNA, and it opens up, okay? Like this, a DNA polymerase, all right, which would come in here and bind here, it has nothing to go off of. It needs that 3' OH. Okay? So in order to provide a starting point, there's a certain primer that you have to do. Right? A certain push. Okay? How many of you ever started a lawnmower? Lawnmower, snowblower, whatever it might be. What do you have to do to the engine before it starts? There's a little bubble on the side of the engine, right? A little plastic bubble you push, you prime the engine, right? You put a little bit of fuel into the carburetor so that when you pull the cord, it starts up right away, okay? You're giving it a starting point, right? And that's what this primer is. It gives a template for the polymerase to bind to and start from. But there has to be something that produces the primer, okay? And this brings in another enzyme, primase. Okay? There's a short RNA primer that is put before DNA polymerase starts. Okay? So we can remove here, let's see, erase. I'm going to erase the DNA polymerase. and we'll put in our RNA polymerase comes in here. There's a, not RNA polymerase, primase comes in here. Okay? And we'll produce an RNA primer, which then leaves that 3' OH. Okay? So a short, maybe 5 to 10 nucleotides RNA primer that will provide a 3' OH that DNA polymerase can then recognize and begin producing DNA. What do you think is the importance of having an RNA primer versus a DNA primer? Why RNA? Sorry, so RNA, well, it's going to bind to DNA though. So it's going to come in and be a double-stranded portion of this. Why is it important that we have RNA nucleotides brought in there and not DNA nucleotides? What's that? All right, so it's going to be in the reverse.
So that was the new
What's going to happen to that RNA later? Is that is that RNA structure going to stay in the DNA that's going to be passed on? Out. So the RNA won't be replicated. What's going to happen to that? Because all right, so if we start with an RNA primer and we continue on, at the end of replication, we're going to have a strand of DNA that's going to look like this, right? We're going to have this, and say we're going to have this other one, and then an RNA primer, right? That little portion is going to be RNA. Is that good or bad? What should a full copied piece of DNA look like? Double-stranded from end to end, right? With nucleotides that are made up of ATGC. What could that RNA have? Could have a U. Okay. What sugar is present in those RNA nucleotides? Ribose, not deoxyribose. So one of the reasons for having RNA nucleotides there versus DNA nucleotides is that they are going to be removed later. All right, so they're going to be removed. We're going to erase those and we're going to replace them with DNA. By having RNA there, okay, ribonucleic acid, the enzyme that removes them can recognize them as ribonucleic acids and tell the difference between those and deoxyribonucleic acids. So they can be removed and replaced by DNA nucleotides so that uracils aren't present in the DNA. Okay? That enzyme that does this, that puts down that RNA primer, is primase. Okay? Short, 5 to 10 nucleotides long. All right? Now that sets up that free 3' end of the RNA primer and will allow DNA polymerase to go. Okay? There's your DNA polymerase. This is the enzyme that catalyzes synthesis of a new nucleotides, a new strand, based off the template strand. All right? And it's very much a puzzle. Okay? DNA polymerase will bind to the 3' primer, recognize what base or what nitrogenous base is on the template, and then basically just do a does this fit type thing. 1, 2, 3, done. All right? Which one fits? Fine, and we move on. All right? About 500 nucleotides per second in bacteria, and about 50 per second in human cells. Why? That's the rate of catalysis, or catalysis, right? Meaning that's how fast a base pair can be incorporated into them. why is it so much slower in human cells? It's more complex. All right? More complex. Okay? Each nucleotide that's added is added to a free 3' end. All right? So every time we add a nucleotide, we're going to add a nucleotide to the free 3' hydroxy group, the OH, okay? And we're going to add it to the 5' end of the sugar. So 3' to 5', and then it's going to leave you another free 3' hydroxy group. So in a way, DNA polymerase is self-priming after this. All right? Every time it adds a nucleotide, it provides another spot to add another nucleotide. Okay? And so you end up, nucleotides come in as triphosphates, right? So you have ATGC, triphosphate, and the one phosphate stays, the other two are released as what we call pyrophosphate. You see it looks like this. So here's the free 3' OH. Okay, we're going to add, if you count on the 5 carbon sugar, right here is the 5' carbon. So we're going to add from 5' phosphate to this 3' OH, okay, and you end up with pyrophosphate, 2 pyrophosphates coming off. All right, this is the formation of what's called a phosphodiester bond. I seriously, when I write on the iPad, it's like my 3rd grader is writing. Less than she has better handwriting than me at that point, anyways. Emily.
What did you mean when you said you're adding from the 5' to the 3'?
So the way we designate numbers on the nucleotides is like this. So we have a 5 carbon sugar, right? This, we're going to draw a nucleotide. Here's your 5 carbon sugar, and off of the first carbon is your base, right? Whatever it might be, ATGC. So now we can count. We go 1, 2, 3, 4, and then there's one that comes off of here, which is number 5. And actually, this is in an oxygen. Okay? So here's your 5 carbon sugar, and then off of this 5th carbon is your phosphate groups. Okay? So when we say 5', right, we're talking about 5', 3'. It gives you directionality of how that's going to be added. So this is going to be a free 3' OH up here from the other nucleotide, right? Your phosphate is going to be produced, your bond between the OH, that OH group, that hydrogen is going to come off, to the phosphate, and then to the carbons of the next nucleotide. All right, so, and then here you have a free 3' OH that's going to be added for a spot for the next nucleotide to be added. Okay? Questions on that? All right. So that's the basic of replication. All right, that's how the process occurs. We're going to start adding nucleotides. There comes a problem when we talk about the anti-parallel nature of this. All right, because the 2 strands are in opposite directions, there is a change. There's a change in the structure, which turns into a difference in the function of replication. Okay, so because the anti-parallel nature of DNA, okay, it will actually changes the way replication occurs on one strand versus the other. All right? So if we look at DNA like this, and we say this is the 5' to 3' direction, right? And its template strand would be what?
3' to 5'.
All right, 3' to 5'. Okay? Replication occurs on the 5' to 3' direction easy, right? Because on this strand, replication is going to occur very easily because what's going to happen? What are the primers that are going to be produced on these two strands? On this strand, it's going to be 5' to 3', right? What is a primer that's going to be produced on this strand? 3' to 5', right? So that can elongation occur in this direction on the upper strand? No, right? It can't occur very well because it doesn't have a free 3' OH. Okay? On this strand down here, okay, elongation can occur very easily because they have your 3' hydroxy stick, right? The OH group. So this is what's called the leading strand. All right? So the leading strand template is in the 3' to 5' direction, and synthesis of the new DNA occurs in a 5' to 3' direction. All right? The leading strand of the template is the 3' to 5' direction. Okay? And synthesis occurs in the 3' to 5' direction. All right? And you see that's kind of written there. DNA polymerase adds nucleotides to the free 3' end of a growing strand. Therefore, a new DNA strand can elongate only in the 5' to 3' direction. So what does that mean for the other strand? Everyone's doing this. It happens in sort of steps, and that's what's called the lagging strand. And we'll go through that. All right? So along one template strand, the DNA polymerase synthesizes in a 5' to 3' direction, all right, continuously. All right, well, what we call is processive. All right, meaning it can add nucleotides continuously, always moving toward the replication fork. Okay. So here is your so there's the template strand, 3' to 5' up top. Okay? And synthesis is occurring 5' to 3' towards the replication fork. So as fast as helicase can open the DNA, the polymerase is putting down, synthesizing the new daughter strands. All right? The question then is, what happens on the other strand? All right? And then also, what happens in the other direction from the origin? Because up here, in this part, you see that I told you that from the origin, replication occurs in both directions. Okay? So in both directions, you're going to have one side be a leading strand, and on the other side is going to be a lagging strand, and then go in the other direction, the opposite is going, the vice versa is going to occur. All right? What's leading on one way is going to be lagging on the other way. Okay? So how do we replicate the lag? All right, so here's your model, leading one way, lagging the other. Right? Because we're always moving towards the replication forks. To elongate the other strand, the lagging strand, DNA polymerase must work in a direction away from the replication fork. And at first this is going to be like, well, why doesn't DNA polymerase just have the Okazaki fragment work in have a 3' OH and work another copy of the DNA just work away from the replication fork and go that way. Well, that was away from the the replication fork. That was a big question for researchers, and it actually turned and sort of adjusted the model about how replication occurs, and you'll see that in a little bit. You'll see that one strand is kind of stop and start, okay, and that's why we call it the lagging strand. The lagging strand is produced in small parts, called Okazaki fragments. All right, named after the Japanese researcher that found them years ago. Okay, so it sort of does one part similar to the leading strand, stops, and then does the next part. Okay, off another arm. All right, and then after, you'll see what the Okazaki fragments look like in a second. What's the problem with doing or replicating DNA in chunks like that?
They need to be joined.
Because they need to be joined, right? There's several RNA primers that are part of a small part of the DNA that need to be removed, and then those chunks need to be joined together, all right? And DNA ligase is an enzyme that will fuse DNA chunks together. All right, so they will ligate them. All right, so here's the lagging strand. RNA primer. All right, so you have RNA primer, 5' to 3'. And then you get DNA polymerase 3, makes Okazaki fragment. All right, and it will continue until it runs into a primer ahead of it. Right? So then it stops. Well, as what's happening to the replication fork as this occurs? DNA polymerase is moving away from the replication fork, and helicase is opening up more DNA in this direction. Right? So now DNA polymerase 3 goes back, has another RNA primer to build off of, and produces another Okazaki fragment until it runs into the preceding RNA primer. And then it gets knocked off and goes back again. So it's very, what we call spasmodic, stop and start replication. It is not continuous like the leading strand. The lagging strand DNA synthesis is discontinuous. All right? Has breaks. And then ligase will come in, right? So you'll notice that DNA polymerase 1 replaces RNA with DNA. Right? So polymerase 3, one DNA polymerase is responsible for basically synthesizing the DNA in a processive manner, meaning it goes and just starts reading and will continue to add base pairs as long as there's a template. Okay, and there's nothing in front of it. DNA polymerase 1 is not processive. It only does a short part. Okay, that's responsible for replacing the RNA primer with DNA. And then DNA ligase will join the nicks between the strands. And then Ben, back to your question at the beginning of this class, DNA ligase also repairs those nicks in the topoisomerase, from what topoisomerase does earlier. Okay? Questions on the lagging strand?
Where does the continuing RNA fragments coming from? I know it has the primase, but why does primase keep making new RNA fragments?
So as the replication fork moves, all right, so if this kept going, you're going to get a continuous chunk of single-stranded DNA. All right, because it's moving away from the replication fork. So primase recognizes that single-stranded DNA and will make an RNA primer. All right, on both sides. It doesn't distinguish leading strand versus lagging strand. It just says, here's single-stranded DNA. I'm going to make a primer for it, regardless. And then, DNA polymerase can come in and do it that way. Okay, so it's there. The enzyme's there, and it will make a primer as long as there is single-stranded DNA available, of a certain size available to it. Okay? So here's just, we go through the primase, right? Pol 3 makes a fragment, right? And then the Pol 3 runs into the preceding Okazaki and then comes off, goes back. Okay, so this is your overall model of leading versus lagging strand. All right, helicase opens the DNA. All right, you have RNA primer on the leading strand. RNA polymerase 3 continues processively towards the replication fork. While on the lagging strand, RNA primase puts down RNA primer sort of at even spaces, and DNA polymerase synthesizes new DNA. That RNA is removed by DNA Pol 1, and ligase joins the fragments together. Eventually, you end up with 2 complete new copies of the DNA. All right, which is semi-parental DNA. All right, so here's all the proteins at the replication fork. All right, single-stranded binding proteins, primase, DNA Pol 3. You have the helicase that's not showing your topoisomerase ahead of it. All right, this is what's going on the lagging strand. DNA Pol 1, DNA ligase is also present. Okay, so really what you have, and I've sort of broken it up into sort of what happens along the replication fork. That's not really what happens inside the cell. Inside the cell, all these proteins are sort of jumbled together, and they form what's called a replisome. All right, so it's called a replisome. So the book calls it a DNA replication machine, a DNA replication complex. All right, it's all the same thing. It's a collection of the proteins that are important for DNA replication to occur in one area, so that we're not waiting for an RNA primer to come in, or primase to come in and go out, or DNA polymerase. They're all there in one area. All right? And the way that there's an updated model about how this occurs, where basically RNA or DNA is pulled into the replisome and sort of pulled through. So if I was to say sort of kind of like 2 strands of a rope that you pull through a pulley system and separate the strands apart. Okay, and as you come apart, you're now, as it comes out, the one strand is being replicated continuously. The other strand has to come through at chunks. And this is what's called the trombone model of DNA replication. All right? And this is sort of what it looks like. All right, so you have DNA Pol 3, your parental DNA, it's sort of pulled through. You'll note there's a couple copies of DNA Pol 3. All right, the helicase enzyme is sort of the middle of all of this. It's going to open up the DNA, and so this being your lagging strand can only be copied in 5' to 3' direction, and it goes through in a certain at certain chunks. All right, this would be important for you to see in animation. So with that, I'm going to